Hi there, folks. Once again, Cassandra Swett, UC Davis Plant Pathology. I'm outside for this presentation with some tomatoes, some nice dead tomatoes in some cases, because I'm going to be talking to you about diagnostics of diseases in tomato, and apologize for any background noise. So we've been working for the last several years to continue to provide services and do research to help you guys make informed management decisions and provide accurate information on pathogens present across the farms in the state. We've been conducting ongoing disease diagnostics activities every year. We receive between about 200 and this year, 120, and this year about 170 tomato samples from across the counties in the state, diagnosing a wide range of diseases. And we also have been managing for Fusarium wilt resistance breaking by monitoring for that dreaded Fusarium wilt race 4. So this year, for example, we recovered about 15 FOL isolates from 2020, as well as 3 putative from 2019 that we're going to be testing for race 4. And diagnosis of these diseases isn't easy. This is a very dead tomato plant, as you can kind of see, but what killed this plant? There's a lot of things that can cause disease in tomato. And one of the kind of groups of culprits are these Fusarium diseases. What's hard about the Fusariums is that they're really hard to tell apart, and we need to tell them apart in order to use things like cultivar resistance and even chemicals and crop rotation. Right now, even our lab-based tools do not work. So we want to develop some informative molecular-based diagnostic tools, some simpler ones, and also some ones that folks like the farm advisors and others across the state can use out in the field. And then, in order to actually do this work, we're proposing to continue some of the funded efforts that we've been doing over the last few years to identify more specific gene regions for Fusarium wilt, as well as for this new emerging issue that we'll be talking about, Fusarium falciforme. And so part of what we're proposing in the early work here for falciforme is we don't even have a way of saying this is falciforme, right? That's the problem. So with Joana del Castillo, my project scientist, we're developing the phenotyping methods that can definitively tell us something is falciforme, and also be able to just differentiate between potentially other Salmonid pathogens in the state. And so the additional issue with us is that, here I am, tomatoes in the background, diseases in this field, what's happening here? I would have loved to know that my plants were gonna die before I planted tomato. Soil testing can do that, but there's not great soil testing tools. Fusarium wilt race 3 is the main soil testing target for the work we're proposing to do. This is also continuing work, doing a soil sampling strategy work with Kelly Paw to figure out how much of my field here should I have sampled the soil from it, and is there a strategy I can use to do that? And then in the lab, are there methods that I could use to more accurately detect the pathogen in the soil, and then combining that with those efforts to develop better molecular regions for quantifying this pathogen in soils. So this is some of what we're proposing to do. And then the last part is, for the time being, there's some methodologies that we want to put out as beta testing tools where we actually plant tomatoes in infested soil. We tested this out last year. It actually seemed to work for the 2 fields we tested. One of them, we got Fusarium wilt in the tomatoes we planted in that infested soil. So we'd like to get samples from you. Please send us samples. We want 5-gallon buckets, and just email me and we will test your soil and tell you whether or not we can detect Fusarium wilt. So this is the breakdown of the total budget that I'm asking for the Swett Lab. If this is not the last presentation, then I'll be presenting on some of this other work. And if it is, this is the summation. And one of the things that I really want to highlight is that, we've got a lot of collaborative projects this year and some excellent leverage cost share dollars. We've been able to bring in a lot of support, $330,700 just for 2021 research. And these are a summary of the leverage support. So these are just some of the outcomes that we generally are aiming for: long-term data sets, enhanced capacity of projects, services that don't otherwise exist, and pushing the envelope and getting future funding. And so we really try to take I try to take a leadership role in these regards. So that's all I've got for diagnostics. I hope you guys have questions, and I look forward to talking to you later.